Good afternoon, everyone. A very warm welcome to all of you present. Thank you for joining us today. I am Yashasvi from the Sequencing Services team at HaystackAnalytics, and I'll be your moderator for today's session. We're delightful to have you with us for this webinar on advanced metagenomics, from resistance cassettes to new frontiers. This webinar is a part of our ongoing efforts to create meaningful scientific conversations around genomics and its expanding applications across research, public health, and clinical ecosystems. For those who are joining us for the first time, Haystack Analytics is a health tech genomics company based at IT Bombay with the mission to bring genomics to the forefront of healthcare. One of our key focus areas has been infectious disease genomics, particularly tuberculosis. Our TB genomics diagnostic solution has been recognized as a top innovation by BIRAC and was launched by the Honorable Prime Minister of India, which has been a major milestone for us. Our flagship product, Infection, is designed to support personalized therapy in infectious diseases and aligns closely with WHO's Sustainable Development Goals, especially in making advanced diagnostics accessible and scalable. Our focus is to fill critical gaps in healthcare by building scalable genomic solutions that benefit patients, clinicians, researchers, and the larger healthcare ecosystem. Through initiatives like today's webinar, we aim to bridge cutting edge research and practical implementation, fostering collaboration across academia, industry, and public health. Looking into our journey so far as an institution, we started off in 2019, where Haystack won Maharashtra Startup Award and the company was incorporated. In 2020, our TB genomics drug resistance test got validated with NTEP, and genomics platform development started. Moving ahead in 2021, Haystack launched its TB Genomics Pan India, being world's first nationwide deployment of TB genomics across private healthcare. In the same year, we also worked on COVID genomics service for BMC, where we predicted Delta variant as VOC to BMC. Moving forward to 2022, which was a remarkable year for us, in which we got BIRAC Big Grant for TB for developing tuberculosis genomics for the National TB Elimination Program. It was the year the development of our universal infectious disease test started, and also Haystack Genomics platform was integrated and used by Intel and GE Edison, which are HIPAA and GDPR compliances. So in 2023, we moved ahead with our universal ID test and achieved POC. It was also the year in which world's first distributed genomic solution for near to point care detection of all bacterial and all fungal infections, we were recognized as that. In the year 2023 itself, Dr. Velumani and other experienced healthcare veterans invested seed capital into Haystack, and our universal ID won at CAHO. In the year 2024, we started off by winning FICCI HEAL 2024, Digital Transformation Initiative of the Year. It was also the year when we were awarded as the Gold Health Tech Solution and Silver Health Tech Startup by FE Healthcare Awards 2024. In the year 2024 itself, we were showcased at the 10th Nat Health Arogya Bharat Summit. Moving forward to 2025, we recently got accredited by NABL and we completed for all our labs in Mumbai. We were also honored with two prestigious awards at TICON 2025 in Boston, USA, where we were winner in the life sciences category and also the overall winner in genomics. Now we'd be introducing our speakers for today. It's my pleasure to introduce our first speaker for today, Dr. Amrutraj Zade. He heads research and development at HaystackAnalytics. Dr. Zade has extensive experience in applying metagenomics across diverse research domains and a focus on translating complex genomics data into actionable insights. He has done his PhD in biotechnology from IIT Bombay itself, and his domain expertise spans across genomics and NGS technologies, molecular diagnostics, and precision medicine, and he has also extensively contributed to research publications and contributions. Our second speaker for today is Dr. Rakeshkumar Yadav, who has done his PhD in advanced research training in microbiome, microbiology, and next generation sequencing disciplines. He also has postdoctoral experience in international academic research in Europe. His domain expertise involves next generation sequencing and genomic data analysis. He's worked extensively on microbial ecology and microbiome studies and applied microbiology and research methodologies. He has also been an active contributor in research related to microbial genomics and ecology and have added his esteemed contributions to scientific research initiatives during postdoctoral tenure at a leading European research institution. Before we begin, a few quick housekeeping points. This, the session is being recorded. Please keep yourself on mute during the session. If you experience internet connectivity issues, kindly wait patiently or rejoin. You can post your questions in the Q&A or chat section at any point during the session. We'll take them up towards the end during the Q&A session. I'll now hand over the session to our first speaker, Dr. Amrutraj Zade, who will be speaking on expanding scientific frontiers through metagenomics. Dr. Amrutraj will set the stage by exploring how metagenomics is transforming research across environmental, clinical, and translational domains. Over to you, Dr. Amrutraj.
Thank you, Yashvi. Very good afternoon to everyone. Yeah, it is a pleasure to present or kind of start the session for Dr. Rakesh, who has such a wide experience in metagenomics, especially in microbial metagenomics for the ecology. I will be briefly touching upon on how we can expand the scientific frontier using metagenomics. So metagenomics is a culture-independent analysis of collective genetic material recorded directly from environment or host-associated samples, which means that we are taking out certain limitations which are associated with culturing these microorganisms with a defined media or a known media as per the requirement of the microorganisms. I will be broadly dividing metagenomics into three ways by which we can look into it. One is metataxonomics, which is using amplicon sequencing of 16S, 18S, and ITS genes. It has advantages and disadvantages. Advantages mainly as it is fast and cost effective. Fast with respect to the sequencing that we are doing. We are specifically targeting the genes, so we are utilizing a smaller region of the genome that is present in the sample. And cost effective because we are generating a lesser amount of data for that, which means that in less amount of data generated, we can gather more information. There are certain challenges associated with it, which are it does not capture the every gene content that is present in the genome. It also has some amplification bias since we are targeting the specific regions like 16S, 18S, and ITS for the taxonomy. we will be not able to target other genes which are involved in the amplification has a bias towards particular organisms and will not be able to cater to those. There will be a bias. The third disadvantage, as we said, that we have only 16S, 18S, and ITS regions probably, or targeting these regions. We will not be having viruses covered into it because these viruses will not fall into these categories. And the main application for meta taxonomy will be profiling of what is present in the sample, microbial ecology, and probably only rRNA based phylogeny that is possible with this. Metagenomics using random shotgun sequencing and DNA or RNA will also have certain advantages and very few challenges. The advantages are no amplification bias, as we are not amplifying any certain region of the genome, and we will be able to detect bacteria, archaea, viruses, and eukaryotes. We also have capability to analyze the data generated from metagenomics by assembling the genomes, de novo assembly of the genomes. However, there are challenges that it requires high read count, which means the data generated is going to be higher than the meta taxonomy, and it will require reference genomes for the classification that we need to do. Main application for this is profiling, of course, for not only the bacterial and eukaryote population, but all the domains of life. And we will also be able to have a functional genome analysis done. We will also be getting the whole genome phylogeny can be generated from this data. And of course, it has a major role in detection of pathogens. Then comes the third category, where meta-transcriptomics using the sequencing of mRNA present in the sample. The advantage that it gives us is that we will be screening all the active genes and the pathways that are present in the sample. However, the challenges are, of course, as we all know, the challenges associated with RNA, that is mRNA being unstable. And there are in the entire process of doing metatranscriptomics, there are multiple purification and amplification steps that are involved, which might lead into the more noise in the data. The main application is transcriptional profiling of whatever organisms or whatever pathways that are active in the sample. The sequencing technology or the NGS platform that can be utilized is short reads platform and long read platform. Short read platform will impose certain amount of challenges with respect to gaps that are being generated during the data analysis. This can be taken care if we generate sufficient amount of data, which increases the depth of sequencing, and then we can cater this, which means that we will have to generate more amount of data. However, with respect to long read sequencing, we probably will not face such an issue where we will be covering all the gaps through the long read sequencing. I would like to highlight certain points with respect to whole metagenome sequencing versus 16S rRNA sequencing. Of course, both of them have their own advantages and disadvantages, as we discussed just now. However, if we look into 16S rRNA sequencing, it will give us taxonomic profiling and functional inferences of the microbiome that is present in the sample. However, in whole metagenome sequencing, we will not only get the taxonomic profiling and functional inferences, we will also be able to cater discovery of novel microorganisms as we are looking into the whole genomic content of the sample. As well as discovery and subsequently discovery of novel proteins. Tracking the intra and inter-individual bacterial transmission that is being done for the microbiome can also be done by whole metagenome sequencing. The microbiome evolution, where not only the 16S part or the ITS part is being catered for the evolution, the other part of the genome can also be done by whole metagenome sequencing. The association studies of microbial genetic variations between the individuals requires much more depth much more depth about the genome than just the 16S and ITS region of the organisms, and that can be only catered by whole metagenome sequencing. And finally, metabolic modeling of a personal microbiome can be catered by whole metagenome sequencing. The application across research domain, if I could highlight, there are few that I have noted it down: environmental and climate research, human and animal health, agriculture and food system, antimicrobial resistance, wastewater epidemiology. Little broader idea about how we can cater all these things. If I take one such example where crop improvement and sustainable agriculture, if we look into that, we can look into the soil microbiome, which can be beneficial for understanding the health of the soil, and also will result into promoting the plant growth. We can also look into the microbiome of the plants, different parts of the plants, to understand the pathogens that are there which have propensity to develop the disease, and also understand the disease resistance in the crop. If we look into the environment, we can look into the environmental monitoring and bioremediation part, where we can identify novel genes which can remove the organic pollutants and then the toxic heavy metals. We can also identify through the metatranscriptomics that we just discussed, novel metabolic pathways that are present in the microbiome, and then we can deploy that for the bioremediation. Such advancements are also being done using metagenomics in clinical forefront, where the rapid and precise identification of the pathogen can be done, ranging from bacteria, fungi, parasite, and viruses, as well as novel bioactive molecules having unique mode of action, and their gene clusters can be identified through transcriptomics of the clinical samples. With respect to the mitigation of greenhouse gases, we can promote the renewable energy to reduce the resilience on the fossil fuels. And of course, we can also identify novel enzymes from the metagenomic studies. One such example I would like to highlight, which is developed by HaystackAnalytics. It is called infection NGS. In infection NGS, we are kind of catering the medical microbiology, removing the biases or the limitations that are there for culture. When we look at the sample, we can directly address the microbiome that is present in the sample and identify the pathogen from that sample. So we do not, as I mentioned, we remove the dependency of culture, which means that the turnaround time for the test reduces significantly. Also, the dependency of the defined media or the fastidious organisms that are there in the sample, which generally do not grow in the routine media culture, we can cater to those identification of those organisms. The process is simple. The sample is collected, the nucleic acid is extracted, and then pre-processing, which is library preparation, sequencing, data analysis, and report, which is done within 24 hours. We have our proprietary universal ID kit, which is also now CE-IVD approved. The data analysis is also automated using Omega ID platform, and it is world's first distributed clinical microbiology genomic software, which can be deployed at multiple centers, which means that the distribution of genomics or metagenomics per se can be achieved at multiple centers across the globe. That is what we have done in India. We are operating at multiple centers in India. We have done around 6,000 plus samples. We have addressed around 15 plus sample types, and we have done around 2,000 plus samples till date. What we have learned from that is globally, even when we look at the WHO guidelines, the focus is on escape pathogens, which is from our data, only 2% of the pathogens that we have detected in the samples. 98% of the organisms that we have detected in the samples across India are non-escape organisms, which means we are looking at the only tip of the iceberg. And that is what a limitation of the culture technique is. and we can actually cater more than that when we look into the metagenomics. If I look into the data across the different sample types, WHO priority pathogens are only contributed ranging from around 5 to 12% across different samples, such as BAL, CSF, plasma, pus, tissue, urine. And not only that, if I look at different sites, the escape pathogens are distributed differently, which means that different sites or different etiology or different location contributes different type of pathogens in India because of course, different food habits and different ecology that is there in the different part of India. We are not only looking into the organisms, we are also catering to the antimicrobial resistant genes. If I look into the distribution of antimicrobial resistant genes, we have not seen that we have seen that the antimicrobial resistant genes are not only limited to escape pathogens, but non-escape pathogens also cater in antimicrobial resistant genes, which means that knowing non-escape pathogens through the methods that are there in the current clinical setup are not going to address the resistance that is there in the system, which means that it will not be able to, the clinicians will not be able to treat the disease in a proper way. This is the core team. We have expertise across clinical microbiology to genomics to human genomics. And of course, we have extensive experience in the bioinformatics as well as the assay development. Our founders are ex-IIT, ex-University of Oxford and ex-University of Denmark, which is Dr. Anirvan Chatterjee. Mr. Gaurav Srivastav, who is co-founder and CEO, he's ex-IIT Kharagpur and has 15 plus years in finance. We also have Professor Kiran Kundabagil, who is our co-founder and he's our scientific advisor. He's professor at IIT Bombay. Our CTO has extensive effort in extensive experience in clinical software development. We have Dr. Mahua, who is Director Medical Affairs, MD Microbiology from PGI Chandigarh. We have Dr. Aparna, who is Head of Growth and Scientific Support for, we have Ms. Shashi Shah, who is Bioinformatics Head. And that's about expanding scientific frontiers through metagenomics. Thank you, everyone. I would like to hand over the session to Yashashvi now.
Thank you, Dr. Amrutraj, for that insightful overview and for setting the stage so clearly on the breadth and impact of metagenomics across research and applied sciences. Building on this broader perspective, we'll now move into a more focused discussion on antimicrobial and resistance gene dynamics. Our second speaker for today is Dr. Rakeshkumar Yadav, a postdoctoral researcher in the Division of Soil and Water Management at KU Leuven, Belgium. Dr. Yadav's work focuses on understanding antimicrobial resistance in environmental microbiomes and the role of integron-associated resistance gene cassettes in resistant dissemination. Today, he'll be presenting on integron-associated resistance gene cassettes, long-range amplicon sequencing approach, where he'll share how long-range amplicon and metagenomic sequencing across diverse ecological samples helps uncover novel resistance cassettes, reservoirs, and transmission pathways, offering valuable insights for resistance surveillance and risk assessment. With that, I invite Dr. Rakeshkumar Yadav to take over the session. Over to you, Dr. Rakesh.
Hello. Yeah, am I audible? Yeah. Thank you for your kind introduction and thank you for the invitation. Do I have the control for the slides?
Rakesh, you are not audible.
Hello. Am I audible?
Yes.
Yeah, okay. So thank you for the kind introduction and thank you for the invitation. So I am Rakesh Kumar. I'm working as a postdoctoral researcher at KU Leuven with Professor Derek Springal, and I'm working on a project which is called as Marshore project, which basically aims at refilling or recharging the groundwater reserves. So it's a multi institutional project. We have several collaborations with different European universities and a company. So as the title suggests, today I'll be discussing about what are the roles of integrons in antibiotic resistant dissemination and development. And I'm also going to discuss about how long range amplicon can be used for understanding these integron associated antibiotic resistant gene cassettes. So yeah, so just maybe can we yeah. So I'm getting a lot of chats here. It's disturbing me, sort of. Yeah. So let's talk briefly about antimicrobial resistance. Now, what are antimicrobial resistance? Antimicrobial resistance simply means that the pathogens do not respond to any clinical treatment because they have developed an ability to inactivate or modify antibiotics. We all understand the gravity of antimicrobial resistance leading to higher mortality and higher economic losses, but it also has a several ecological consequences. For an example, when we take antibiotics, around 60 to 70% of the antibiotics are not metabolized by the body, and it is directly released into the environment. Likewise, there are different sources of antibiotics and antibiotic residues which ultimately ends up in the environment. Now, what happens when they go in the environment? Environment act as a sink. It collects antibiotic residues and pathogens from different sources, and then what happens there is it provides an optimal niche for further dissemination of antibiotic resistance. Now, how does this happen? It happens mainly because of the two reasons. The first is mutation. So during the stress conditions, these pathogenic microorganisms develop a lot of mutations, which helps them to survive these antibiotics. And the second important process which helps in development of this antibiotic resistance is the lateral gene transfer, wherein unlike in mutation, where the mutation is passed to the descendant, in lateral gene transfer, the mutation is transferred from one bacteria to another, also from different species of bacteria. So the bottom line is we humans and our human activities are generally contribute to ARG's dissemination and development, but the major facilitator is are the mobile genetic elements. Now, as the name suggests, these are the genetic elements which can move from one genome location to other genome location or from one bacteria to another bacteria by various processes, which includes transformation, conjugation, transduction, and etc. etc. So how I cannot move the slides. Sorry. Yeah. So these mobile genetic elements are basically selfish entities. So what I mean by selfish entities is basically they use the host machinery to replicate themselves, to propagate themselves, and then even in some cases they can also kill the progeny cells which do not have mobile genetic elements. Further, they are very dangerous pollutants. That means they are not like chemical pollutants which can be removed or which can have some restricted usage. These are pollutants which can replicate and amplify, and it's a never ending pollutant. And because of these characteristics, they have become invasive species. For example, they start with genes, then they colonize different microbial communities and ultimately colonize the entire landscapes. So there are different types of mobile genetic elements like transposable DNA elements, which can jump from one DNA element to another DNA element. There are conjugative DNA elements, which can transfer between two different bacteria. But our focus is integrons, so I'll not discuss the other mobile genetic elements, but we will focus on integrons. So let's talk about integrons. I cannot move my slides. Yeah. It's a bit slow. It's lagging. Uh,
Rakesh, I'll do
so
that for you. Sorry. I
yeah, okay,
Yeah,
okay,
you can
fine.
just say next. I will do it for you.
Yeah, so let's talk about integrons. Now, integrons, think of a smart investors. So what do the smart investor does? It maintains a very diverse portfolio of shares, and according to the changing conditions, it will remove some of its shares which are loss making and acquire some newer shares which will give them profit. And also it will, the smart investor will try to rearrange their portfolio so that it is always profitable. Think of integrons as a smart investors. So integrons, I'll tell you why. You will see it in further slides how it is a smart investor. So that there are three main features of integrons: integron integrases, which are basically a recombinase enzyme involved in recombination, and then there is a recombination site, which is ATTI site, wherein the recombination of the incoming gene cassettes takes place, and then there is promoter, which basically drives the expression of these ORF cassettes. Now, class 1 integron. So there are many different classes of integrons depending upon the integrase enzymes, depending upon the sequences of the integrase enzymes. But among all the classes of integrons, class 1 integrons are most dominant integrons because of their several characteristics. For example, they are quite flexible in recognizing different ATTC sites, which acts as a substrate for integrase enzymes. Additionally, unlike other integrons, they do not require any bacterial species specific factors for their integration. So class 1 integrons are one among the most dominant. Can you next? Yeah, and then when you talk about the prevalence of these integrons, as you can see on the right hand side of phylogenetic distribution, it is present in among almost all the bacterium phylum. Obviously, it is absent in some of them, but they are diverse and their range is diverse, and mostly they are quite prevalent in proteobacteria, especially gamma proteobacteria, which has most of the escape pathogens like Dr. Amrut was talking about. So, but obviously it is not only restricted to apps escape pathogens, but they are also present in other bacterial population. And like the latest trend was around they are present in almost 15 to 17% of known sequence bacterial genomes. And there was one study wherein around 80 clinical strains of enterobacter was isolated from urine, and the class 1 integrons was present almost in 38% of strains. And if you go by their abundance, almost 10 raised to 23 copies of integrons are released per gram of feces every day. So it's a huge prevalence of integrons. Next. Now we know about what is integrons. Now we look into how do they function. So if you see this circle with ATCC site, this is nothing but a gene cassette which has one ORF and a recombination site which is known as ATTC site. Now these ATT sites are basically recombination site. It has a palindromic sequence, so it can form this secondary structure, so it can go and dock itself in the recombination site of integron, which is at I site. Once, so this integration is basically mediated by the integrase enzyme, which is present in the integrons. And then once it is integrated into the integron platform, this promoter drives the expression of these ORF, which is integrated. Next. Let's go into further details of how integrons work. Now, integrons mainly work with three principles. So basically, they sample and capture different ORF, then they integrate and recombine this ORF into their platform, and then they express the phenotype and they check the phenotype whether the phenotype is good or not, and then they decide whether to keep or not to keep these ORFs. So what happens is integrases generally are inactive, they are not expressed, But whenever there is some stress to the microbial cells, for example, let's say there is a change in pH, there is a change in temperature, or if there is some antibiotics are present, there is this SOS response is generated. Because of this SOS response which is triggered, it triggers the expression of integrases enzymes from this part. Once the integrase enzyme is expressed, they sample among different gene cassettes, and with the help of ATTI and ATTC sites, this gene cassette is basically integrated into the integrons. Now, the next step is once integrated, it will express these ORF, which will apparently code for some protein using the promoter, which is a very good promoter, and it will test the phenotypes. If the phenotype is good for the integrons, if it is good for the microbial cells for its survival, it will keep the gene cassette. Otherwise, it can also remove this integrated cassettes again and release it back to the environment. Likewise, it will go on integrating various gene cassettes as long as the condition is not suitable, and then as long as it doesn't get any right fit for its survival. Sometimes they also rearrange these gene cassettes because the gene cassette which is far from the promoter has a very less expression. So in this way, they maintain a very diverse pool. So you remember I was giving an analogy to smart investors. Integrons are really smart investors because they 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 collect the gene and they express and they they see that whether the phenotype is profitable or not. Next. So now, once we know about the integrons, now we take a step back and try to understand what I am doing here. So we are working on a Mars system. So as you all know, there is the freshwater. There is dismal condition of all the freshwater resources worldwide, and then there is increased reliance on the groundwater, and there because of this increased reliance and because of some other factors, the groundwater is depleting. So there is a need to recharge this groundwater table. Next. So we are working on a concept wherein we are trying to utilize some surface impaired waters. For example, let's say wastewater effluents. So we will use this wastewater effluents to recharge the aquifer, and as a result, the groundwater level will increase. Now, this is not as simple as it sounds, because these surface impaired waters such as wastewater effluent contains a lot of trace organic chemical pollutants and also contains several pathogens even after treatment in wastewater industry. So we need to make sure that before it is used for recharging the groundwater reserve, we need to treat it again. Next. So that is why what we do is we collect different, so our collaborators basically collect different sources of wastewater effluents, and then we use some advanced pre-treatment methods like ozonation, ultrafiltration, nanofiltration, and sometimes we combine all the pre-treatment step. Once the water is treated, it is then used to recharge the aquifer. When I say aquifer, just imagine that it's a ground, it's a soil level wherein the water goes through the ground and filters. So in order to mimic that aquifer in the lab, we have this column system. uh, if you see the diagram below the schematic, which is packed with sands. So these are sand columns which basically resembles this aquifer system, and we also have a field level experiment ongoing. So once the pre-treatment is done, the water is passed to these sand columns. So the water will filter from these sand columns, and then we collect different samples to analyze. So you must be wondering what is the role of integrons, or what is the role of microbial community here. So coming back to again the schematic. So once we collect the wastewater treated effluent, mainly the secondary effluents, we do pre-treatment using various advanced pre-treatment methods. We sometimes also utilize certain other strategies like biostimulation and bioaugmentation. For example, when we add some nutrients and we add some microorganisms to promote the degradation of various trace organic chemical pollutants. And While doing so, what we do is we collect sample coming out of the pre-treatment, and we also collect samples from the sand columns, and we collect the final outlet water, which is then will be used for groundwater reserve. And then we analyze different advanced parameters, which includes physical, chemical, like we measure certain trace organic chemical pollutants. And our role is basically focused on microbiology of these samples. So the idea is we collect samples, we do sequencing, we try to understand the microbial community structure and stability as a function of different pre-treatments and as a function of time. And then we also look for certain ecological functions, like whether there is some degradation function, whether the community is stable or not. So ultimately we want to validate our pre-treatment, and we have to select best pre-treatment which is positive towards the pollutant degradation and negative towards antibiotic resistant genes. Next. But there is one more problem related to this. Now, as I said, we are using wastewater treatment treated effluent. So they are very low in biomass. So you do not have a lot of DNA coming out of these wastewater treated effluent. Further, we are again subjecting this wastewater treated effluent to various pre-treatments. Again, it is reducing the biomass. So how do we do it? So uh, doing direct whole metagenomics sequencing, wherein you just extract DNA and sequence the DNA, is not possible because there is low biomass, and then there is low DNA. So what we do is we use another strategy. We use long range PCR. Now, what is this long range PCR? So we basically design a primer in such a way for various mobile genetic elements which are known to encode for antibiotic resistance or different pollutant degrading functions like integrons and IS elements. So we design a primer in a way that it could amplify all the cargo genes which present between the two primer binding sites. So it can range from anywhere from 10 kb to 20 kb, or even if more, if you successfully extract high molecular weight DNA and do successfully PCR. So this is basically a long range PCR wherein you try to amplify whatever the cargo genes present between these mobile genetic elements, and then we go for sequencing. Let me clarify again. So we go again. So next. Yeah, so let's try how it works. So let's say I have a sample which is in low biomass, which has a low DNA concentration. We try to extract high molecular weight DNA. So when I say high molecular weight DNA, it means that we need to extract a longer DNA fragment, because if we don't have a longer DNA fragments, we don't have a big gene cassettes of functions which are encoded by these primers or these mobile genetic elements. So once we extract high molecular weight DNA, we have, thankfully, for integrons we already have several primers in the literature. But for other mobile genetic elements like is elements, various is elements, we need to design a primer. For example, if you see this is diagram here, we design primer in such a way that everything encoded between the two is elements are getting amplified. So we need a bigger amplicon so that it can span several genes. Next. So once we do this, we do PCR and then we run a gel to confirm whether there is amplification. You will see these variable kind of amplicons because some gene cassettes are smaller, some gene cassettes are larger. So accordingly, you will see a lot of variability in the length. Next. So now once we have these amplicons, we do this, we basically use nanopore sequencing to sequence these amplicons because of few obvious reasons. For example, nanopore provides long reads. So it is essential to have long reads because these mobile genetic elements frequently contains repetitive sites. If you use some Illumina sequencing or some short read sequencing, what happens, these repeats are basically collapsed because the read length is generally smaller than the repetitive sequence. So the sequencer doesn't understand whether the gene is coming from two different ORFs or it is from the same. So it considers one ORF and it's kind of collapses the repeats into ones. So that is the reason, that is the most important reason uh, why we use MinION based nanopore sequencing. So once we do the sequencing, we have various long amplicons of varying sizes. Next. And then we do some initial pre-processing of the sequencing data, and we try to select some try to select reads with highest quality. And now here we used a cluster based annotation wherein we cluster these reads into different clusters as per their sequence similarity and the sizes. We try to correct these clusters because of the inherent nature of error prone reads from nanopore. We try to correct these clusters, and then we make consensus out of these clusters, taking the representative reads from each clusters and then predicting ORF, and then doing further annotations, especially for antibiotic resistant genes and functional genes. So in short, next. So as you can see, I'm going to explain few of the results which we got. So here, this is from one of the institution wherein we were working with different pre-treatment. So we had this inlet sample which is going in the column, and then we have outlet sample which is coming out of the column. So the inlet sample is basically pre-treated samples. As you can see, we detect a lot of antibiotic resistant genes, and especially the antibiotic resistant genes which are well reported for beta lactamase activities, especially to the even the last resort antibiotics such as carbapinoms. Likewise, we detect resistance against different classes. So you imagine you have a very low DNA concentration and still you are getting this many antibiotic resistances, which will not be possible by the way by using a whole metagenomic sequencing approach. Next. And this approach also help us to understand what are the differences between the pre-treatments. On the left you see a graph which is a Shannon diversity indices graph, which basically understands the alpha diversity. When I say alpha diversity, it means that how many different type of antibiotic resistant genes are present in one sample and how abundant they are. So how if there are many antibiotic resistant genes, and if they are abundant also, then the sample is diverse enough. So if you see with respect to control, we see decrease in the diversity of ARGs in all the pre-treatments, especially for the pre-treatment 5, wherein we employ this nanofiltration strategy, wherein we see a lot of decrease significant, although it is not shown in the figure, we see quite a significant decrease in the overall antibiotic resistant genes in the pre-treatment 5. Likewise, the other important thing is we are employing different primers for targeting such integrons. So if you see, we have employed two types of primer, which is one is clinical integron based primers and the other one is environmental integron based primers. If you see this beta diversity plot, you see a strong two clusters. So when you say clusters, the points, basically each points represent one sample, and if they are clustered together, they are more similar in the antibiotic resistant gene content. So if you see the differences between clinical and environment, you see a clear cluster between two primers. What does it indicate? It indicate that we are covering a very good amount of diversity by using this long range approach for integrons. Next. And the findings are not only restricted to understanding what antibiotic resistant genes are prevalent or how much they are abundant. We also try to understand whether these antibiotic resistant genes are present in the same cluster. So if you have more antibiotic resistant genes in the same cluster, that means it is more dangerous, because once this cluster is transferred to some other bacteria, it becomes multi-drug resistant bacteria. So as you can see in controls, we see a lot of clusters, of different antibiotic resistant genes as compared to pre-treatments, but we see a reduced clusters in most of the pre-treatment, especially for the pre-treatment, the last pre-treatment, which is NF based pre-treatment, wherein we don't see any sort of such clusters. So this long read based, long range PCR based approach for integrons helps us to validate which pre-treatment is better. Next. So the previous, yeah. So likewise, we are also working on different insertion sequences, which is another mobile genetic elements. Uh, Currently, we are standardizing some primers for IS-26, which is known to be involved in antimicrobial resistance. Likewise, we also have optimized protocol for various IS elements like IS6100, IS1071, and likewise. Next. Yeah. So whether this study is restricted to only environmental aspect, no, it is very important clinically also. For example, if you see uh, one of the papers by Tavares et al., it says that they detected almost 159 beta lactamase encoding gene cases from all the metagenomes they collected, and which represents all the Ambler classes. That means all the beta lactamases classes, class A to class T, which is a very dangerous situation. Likewise, there was a study, especially the second study, which was related to Indian rivers, where they found that most of these class integrons carry these carbapinamases genes. Carbapinam is one of the last resort antibiotics that is used for treatment of multiple drug resistant infection. Likewise, if you see, it is not only restricted to gram-negative pathogens, but it is also quite prevalent in gram-positive, especially MRSA Staphylococcus aureus. As you can see, almost 78% to 92% of MRSA were integrase positive. Next. And here the last study wherein they try to understand the presence of integrons in sepsis. So a good thing for Haystack where they work on these sepsis infections. As you can see, around 90 to 93% of the detected gram-negative bacteria had almost 31 to 24 to 31% of the uh, gram-negative bacteria were multi-drug resistance. So what does it indicate? It indicates that these integrons are quite diverse. They are not only present in escape pathogens like Dr. Amrut was saying about that most of the antibiotic resistant genes are not only restricted to escape pathogens. So we see it clearly that these integrons carry a lot of diverse antibiotic resistance, clinically very, very important and active against even the last resort antibiotics. And just food for thought is maybe we should start thinking about developing a panel which will have this integron as a primer, so we don't have to selectively target few antibiotic resistant genes that we are currently using. So it would be a good idea to also carry out or make such diagnostic kits. So yeah. So yeah, in summary, I would like to summarize is that these integrons are not only restricted for antibiotic resistant genes. If you see various literature, most of the gene cassettes which these integrons carry are not only related to antibiotic resistant genes, but also related to some other functions. For example, they also give functions such as virulence gene, which is also a dangerous. They also provide some proteins which can inactivate the phages, bacteriophages. So it can also inactivate the phage therapy, which is currently in boom. So these integrons are very diverse, and the gene they encode are very diverse, and basically it's not only restricted to antibiotic resistant genes but also to other functions, which makes these pathogens quite versatile to survive in such clinical conditions. And in last, yeah, antibiotics are not candies, so try to take it more responsibly. Hope you like the talk. Any questions? Thank you.